Thank you very much, Rochelle. Well, good evening, everyone, and thank you for joining Defeat Duchenne Canada for our 2022 research grant announcement. We'll wait just a couple more minutes and begin just after or just at 7 o'clock for people to join. But in the meantime, I'll share a couple of housekeeping points with you. You may have noticed that you've been placed on mute and your video has also been disabled. If you would like to test your volume, please click on the up arrow next to the audio button on the bottom left-hand side of your screen. This will allow you to adjust the volume so you can hear it the way you would like. And while nothing can replace the sense of community and energy when we're all physically together, although that is starting to come back and that is nice to see, we want to try and have as much engagement throughout this evening as possible. So your keyboard will become your voice to connect with other attendees by using the chat function on the bottom. And while we have a packed agenda, we hope to have a few minutes at the end of each of the presentations to have some questions and answers. And you can do that by enabling the Q&A function at the bottom of your screen. Please use the raise hand tool if you need any support from staff members during the presentation. And finally, we will be recording this evening's event and it will be available to everyone early to mid next week. And we'll give it just a few more minutes to get to 7 o'clock and then we will begin with this evening's 2022 grants announcement. Again, welcome to everyone, and if you don't mind, maybe even sharing where you're joining us from this evening, as in the chat feature at the bottom. It's great to see people from across North America joining us for this evening's announcement. This is the third time that we have hosted this event virtually, and hopefully one of these days we'll be able to do more of a hybrid environment, being able to share both in person and virtually with folks across the country and around the world. All right, we'll give it another minute. It's just 7 o'clock now, so sit tight. We will gather here in just a minute and begin this evening's presentation. Okay, it's now 7 p.m. or just a little after, and it's time for this year's Defeat Duchenne Canada Research Grants announcement. Good evening. My name is Perry Esler, Chief Executive Officer of Defeat Duchenne Canada, and it is my pleasure to welcome you to our annual funding announcement. Over the next 90 minutes or so, we'll hear from Nicola Worsfold, our Vice President of Research, Advocacy, and Education, on our research funding priorities and the annual granting process. We'll introduce you to Laurie Power, a Nova Scotia Duchenne mom who chairs our Duchenne Family Advisory Committee, and to Dr. Lisa Hoffman, representing our Board of Directors, and she is also the chair of our Research Funding Advisory Committee. Tonight's grant announcement is the first as Defeat Duchenne Canada. You may remember that earlier this year, we rebranded to better align our cause with mission and our commitment as a national voice for Duchenne muscular dystrophy. From 1995 until earlier this year, you knew us, of course, as Jesse's Journey. And while our name has changed, our vision remains the same: a future without Duchenne. This rebrand wasn't about leaving our past behind. Rather, it was to build on it and create deeper impact in funding research and our support of the Duchenne community. So what is Defeat Duchenne Canada about? As you know, Jesse's Journey was born to focus on research funding, and to date, the organization has contributed over $14.8 million to Duchenne-focused research projects around the world. We are also dedicated to the community through education and support, helping families navigate the landscape in Canada. Advocacy is another important element of what we do to further the Duchenne voice with health and government leaders both nationally and provincially, while advocating for access to affordable treatments as they become available. And of course, we can only execute this work backed by the generosity of our donors. From family fundraising events to generous individual and corporate partners across the world, we are able to move from what to why. It began with Jesse Davidson, and even in 1995, the Davidson family knew it wasn't just about their son, one young man with Duchenne muscular dystrophy, but rather the mission to defeat Duchenne was all about that for boys and young men in Canada and around the world. Boys like 14-year-old Emery in Toronto, Ontario.
I was never one of those little kids who jumped off the back of the sofa. I didn't run or jump like other kids. When I was almost 5, I had a ruptured appendix, which led to a lot of tests. That's when we learned that I have Duchenne muscular dystrophy. It's not easy having Duchenne. My muscles are weaker than my 14-year-old friends. I'm really short because of my steroid treatment. Life is less predictable with Duchenne. But I'm a fighter. Duchenne may limit my mobility, but it doesn't limit my imagination and my determination. And I'm even stronger knowing I have a whole community behind me. My family, friends, researchers, medical professionals, fundraisers, and volunteers. They're fighting for me. They're fighting for all the boys and young men with Duchenne.
Join the fight!
Please donate and help us defeat Duchenne.
And now to share more on our research priorities and our annual granting process, I'd like to introduce you to our National Vice President of Research, Advocacy, and Education, Nicola Worsfold. Nicola?
Thank you so much, Perry, and good evening, everyone. Thank you for joining us today. I'd like to share a few words with you about our research program here at Defeat Duchenne Canada. And we don't do this alone. This is truly a partnership with our scientists, our clinicians, and families working closely together with one common purpose, and that is improving the health and the quality of life of individuals living with Duchenne and Becker muscular dystrophy. And we do this by building a pipeline of new innovative medicines for Duchenne and Becker through the research grant competition. Our key priorities are to fund the most promising research with the best chance of having impact and providing benefit to individuals living with Duchenne and Becker. And we prioritize those projects with the greatest chance of translating to clinical practice in a timely and affordable manner. And I just want to talk a little bit about our process. And hold on, I just can't advance at the moment. It's not working. Oh, there we go. Thank you. Our research granting cycle has 2 rounds to it. And our first round starts in September, where we invite researchers from across the world to submit their letters of intent. We have 2 research committees, our Research Funding Advisory Committee and our Duchenne Family Advisory Committee, that review all of the applications, and they will recommend their top applications that they would like to invite back to a full application. And that happens sometime in the fall, and that starts our second round of our process. And in this second round, we once again review our top applications and our committees. So both of our committees will recommend the top projects they feel that we should fund this year. And that goes to our board of directors for final approval. And I just want to take this moment now to thank our committee members. So our Research Funding Advisory Committee is comprised of researchers and clinicians from across Canada and the US. And we have our Duchenne Family Advisory Committee, which is comprised of parents, sibling, and an individual with Duchenne from across Canada, representing all ages and stages of Duchenne. And our Research Funding Advisory Committee is chaired by Dr. Lisa Hoffman, which is here, who's here with us today. And our Duchenne Family Advisory Committee is chaired by Laurie Power, who's also with us today. And I just want to say that it is the collective effort of these individuals that help us decide where our donors' generous gifts have the most impact on the lives of those living with Duchenne. So I want to thank all of our committee members for donating your precious time to this important process. We could not do this without you. And with that, I would love to pass it now off to Laurie Power, our Duchenne Family Advisory Committee Chair, to say a few words. Laurie? Is Laurie, you're there?
I am here.
Oh, there you are. Okay.
Good evening, everyone. My name is Laurie Power, and I have a 12-year-old son who has Duchenne. And I'm also the chair of the Defeat Duchenne Canada's Duchenne Family Advisory Committee this year. And our committee is made up of families from all across Canada, including parents, a young man living with Duchenne, and a sibling. So we have all, like as Nicola mentioned, we have all ages and stages of Duchenne represented within our group. The Duchenne Family Advisory Committee, working in conjunction with the Research Advisory Committee, reviews all the funding applications received each year. We score the applications and decide which projects will have the most positive impact on Duchenne research. It can often be a very difficult task to decide between so many innovative, intriguing, and promising projects. And I know that our group feels honored to be trusted with helping to decide where the hard-earned funding dollars will be invested each year. Our committee is a voice for all Canadian families living with a Duchenne diagnosis. And as families, we're understandably anxious and for a miracle treatment for something, anything that will help our loved ones retain their abilities, function, and quality of life before Duchenne steals it away. And I know that I personally draw so much hope from all the research being done around the world. And I'd like to thank all of the incredible scientists and physicians who've dedicated their careers to helping find treatments and hopefully one day a cure for Duchenne. And just in closing, I'd like to share a quote that really resonates with me. It's from Dr. Martin Luther King Jr. And I think it perfectly sums up the ups and downs of Duchenne as we wait for all of this promising research to be completed. We must accept finite disappointment, but never lose infinite hope. So thank you again to all of the researchers for being a source of infinite hope for Duchenne families across Canada.
Thank you so much, Laurie. And I just want to say our family committee is something that we've had in place now for the last 2 years. And it's such an important part of our process, having that family input, helping us make those decisions. Because at the end of the day, this research has to be meaningful to our families. And under the leadership of Laurie and our committee, it's been hopefully a good experience for everybody that's done this. And I know I've enjoyed it very much. So thanks so much, Laurie. So with that, we're going to move into our ongoing research highlights. And I first want to start with just sharing with you some video messages from our current grant recipients. So the first ones are going to be our grant recipients that have currently completed their first year of funding and just starting their second year. And we're going to start with Dr. Ronald Cohen, who is the CEO of Hospital of Sick Kids in Toronto, Ontario, Canada. And Dr. Cohen's lab are investigating a novel approach to editing mutation in the dystrophin gene. And the dystrophin gene is, again, that gene that is responsible for both Duchenne and Becker muscular dystrophy. And he's also looking to simultaneously increase the production of dystrophin protein, which is that protein that individuals with both Becker and Duchenne are lacking. So let's hear from Dr. Cohen.
Hello everyone, my name is Ronnie Cohen and I'm a pediatrician and geneticist, and I'm currently the president and CEO of the Hospital for Sick Children in Toronto, Canada. I would like to thank all of you for your incredible support of our research in Duchenne muscular dystrophy. It is support like yours that allows us scientists to pursue exciting outside-the-box research projects that carry a very high potential for changing the therapeutic landscape for boys with Duchenne muscular dystrophy. With your support, we are currently investigating how we can use a novel approach of editing a mutation in the dystrophin gene and at the same time trying to upregulate and increase dystrophin expression. We are very excited about this research, and my graduate student Matthew Rock will now explain our research in more detail. Thank you, thank you, thank you.
Thanks, Ronnie. So hello everyone, I'm Matthew Rock, a fourth-year PhD student in molecular genetics at the University of Toronto, and I'm currently working on this project. So with our work here, what we're really aiming to do is enhance our existing approach for fixing DMD duplications using the genome editing tool CRISPR-Cas9. Now this tool lets us use a protein called Cas9 to cut DNA at a desired spot. And this opens many possibilities for fixing disease-causing mutations. And most importantly for us, this includes DMD. So last year, our lab published work which showed we could correct a very large DMD duplication in mice using Cas9. The duplication was removed as shown here by cutting within the original and duplicated DNA of the dystrophin gene, which removed the now duplicated DNA, restoring the full-length dystrophin. The mice themselves recovered substantial dystrophin protein and showed improved muscle function. However, we didn't see complete disease recovery, and even the highest amounts of dystrophin that we saw were just below the minimum levels needed to fully protect muscle. So we really wanted to improve the strategy and decided to use CRISPR in a totally different way. By modifying the Cas9 and targeting the part of a gene called the promoter, the amount that gene is expressed can be greatly increased, and we call this upregulation. So if at the end of the day, we want to get more dystrophin, why not just upregulate the dystrophin gene itself with CRISPR in combination with our original duplication removal strategy? To do both simultaneously would normally be too complicated to even consider as a therapy, but we have found a solution that simplified our strategy enough to make this entire project possible in the first place. At the beginning of our first year of funding, we had already shown that in mouse cells, we can use a modified Cas9 to upregulate dystrophin. However, this modified Cas9 had more difficulty cutting DNA, so our primary progress since last year has involved much testing to get past this hurdle. We were successful, and even identified a second Cas9 variant that we can use in our mouse experiments. So we are currently on track with our timeline, having wrapped up the first aim of this project and being well underway with our second. Our DMD duplication mice have been given a local injection into their leg muscles with viruses carrying our combined editing and upregulation system. And we're currently determining how much dystrophin was recovered in these muscles, as well as any potential improvements we can make to our strategy. We anticipate this year to move on to our final aim, which is the systemic treatment of our mice with this strategy, which will let us target all the muscles throughout the body. Our greatest hope really is that we will recover even higher levels of dystrophin than we've seen before, which can stop DMD in its tracks and further demonstrate the therapeutic potential of combining existing strategies for DMD with CRISPR upregulation of dystrophin. Thank you.
We will now hear from Dr. Nicholas Bengtson from the Wellstone Muscular Dystrophy Research Center at the University of Washington in Seattle, Washington. Dr. Bengtson is investigating an alternative method of gene editing to restore normal dystrophin production. And again, that dystrophin is the protein that is missing in individuals with both Duchenne and Becker muscular dystrophy. So let's hear from Dr. Bengtson next.
Hi, I'm Nick Bengtson. I'm an assistant professor of neurology at the University of Washington in Seattle. I am currently starting year 2 of my research project that is entitled Development of Gene Regulatory Cassettes that Enable Safe and Efficient In Vivo Dystrophin Gene Editing in Muscle Stem Cells. As this relatively long title suggests, this project aims to establish new technologies that can be used to correct dystrophin mutations in muscle stem cells. And this we would use to improve long-term therapeutic benefits by making sure that dystrophin continues to be produced even if muscle cells are replaced due to normal conditioning or as a result of an unfortunate trauma event. So far in year 1, we have identified several large human genomic regions that are responsible for controlling expression of certain genes only in muscle stem cells. These regions have been copied into smaller synthetic regulatory cassettes that can be used to express genes in muscle stem cell cultures. We are currently in the process of thoroughly vetting several candidate cassettes in cell culture in order to select the best ones for the next step, which will be to test them in our mouse models. So in terms of that, we have obtained and started to expand 2 transgenic mouse lines that will help us specifically research the effectiveness and safety of our cassettes in muscle stem cells on a whole body level. The fine-tuning and optimization of promising cassettes for use in animal models will be the focus of this upcoming year. Finally, I would like to say thank you very much for your continued support, and I look forward to advancing these important studies closer towards clinical application.
Our next grant we will present is from Dr. Sachiko Sato. It's from the University of Laval in Quebec City, here in Canada. Dr. Sato and her group are investigating a new oral administrative therapy to help strengthen muscle fibers in hopes that it will prevent the progressive muscle loss that we see in Duchenne muscular dystrophy. So we'll hear from Dr. Sato next.
Hello, my name is Sachiko Sato. I'm a professor at the Faculty of Medicine, Université Laval, and a researcher at CHU Quebec Research Center in Quebec City. First, we would like to thank Duchenne Canada to support this project and the GlycoNet Center of Excellence of Canada. Together with Dr. Jerome Frenet and Masahiko Sato, we aim to develop an oral therapy using a monosaccharide called N-acetylglucosamine to mitigate the progression of all Duchenne muscular dystrophy patients. We don't have a time to get into the detail about the reason, but we hypothesized that by using 100% pure N-acetylglucosamine, this treatment can create an environment where the muscle can bind more stably to the body to resist the muscle contraction. In addition, we hypothesized that the treatment can optimize muscle regeneration. Our previous results suggest that intraperitoneal administration of N-acetylglucosamine mitigates the progression in a mouse model of Duchenne muscular dystrophy. In this funded project, we like to address whether oral treatment with N-acetylglucosamine also mitigate the progression. Our preliminary result using MDX mouse suggests that one oral 1 month treatment reduced muscle damage and increased the daily locomotion. They also indicate 2 potential optimal oral treatment doses. In addition, our result using a newly developed in vitro myogenesis assay suggests that co-treatment with prednisolone and N-acetylglucosamine synergetically enhance muscle regeneration, implying combination therapy is possible. In addition, we finished 1/3 of our chronic treatment study for 14 months, and we did not observe any adverse effect so far. In summary, our preliminary results suggest that oral N-acetylglucosamine treatment is effective. Please note that effect was confirmed only by the 100% pure N-acetylglucosamine, but not the one found in the supplement quota of a pharmacy. Once proven, this treatment could be taken by all Duchenne muscular dystrophy patients, regardless of their mutations. In addition, we expect that N-acetylglucosamine-based treatment could be combined with other non-targeted and targeted therapies, as the molecular mechanism is likely very different from most of those treatments. Thank you.
And our final project I'd like to show that's in their, or just completed their first year, just starting their second year of their grant, is Dr. Natasha Chang from Mcgill University in Montreal, Quebec, here in Canada. Dr. Chang is investigating compounds that could potentially improve muscle strength and slow down progression in Duchenne muscular dystrophy. So let's hear from Dr. Chang next.
Hi, everyone. My name is Natasha Chang, and I'm an assistant professor at Mcgill University in Montreal, Canada. And today, I'm really happy to be presenting to you our progress on our Defeat Duchenne Canada funded research project entitled Inducing Stress Granule Formation in Muscle Stem Cells to Treat Duchenne Muscular Dystrophy. And so this is a collaborative research project between myself and Dr. Imed Galuzzi. And we just finished our first year of funding in our 3-year funded project. And so the rationale for our project is quite simple. We built on previous work from Dr. Galuzzi's lab, where he showed that in a mouse model of cancer-induced cachexia, which is a severe muscle wasting disorder induced by cancer, he showed that treatment of these mice with a specific class of compounds was able to then improve muscle formation, which is also known as muscle differentiation. And it also improved the strength of these muscles. And so we asked whether or not these compounds could also work in the context of Duchenne muscular dystrophy. And the reason for doing that is that recent research in the muscle stem cell field has shown that muscle stem cells in the context of Duchenne muscular dystrophy are functionally impaired, and they inefficiently undergo differentiation to, and that contributes to the disease and the muscle degeneration that we observe in Duchenne muscular dystrophy. So we asked if we can then improve muscle stem cell function in DMD as a way to help with the disease. And so I just wanted to introduce the key people that are involved in this project. This project is really led by a really talented graduate student in my lab, Romina Filippelli, and she was helped by Shirley Lee. And so what Romina and Shirley showed is that using primary myoblasts, these are muscle progenitor cells isolated from both normal mice as well as our DMD mouse model, which is called the MDX mouse, they were able to show that using varying doses of the compounds that we can then improve muscle differentiation, both in a healthy context as well as a dystrophic context. And so these results were really exciting to us. And now they have validated these results using different variations of this compound. And so what we've now accomplished in year 1, which is our milestone for year 1, is the in vitro validation. So we've shown that in cell culture, these compounds work really well. And we've also started some mechanistic studies to understand what are the key proteins that are involved in this pro-differentiation process. And what we're now looking forward to as we enter into year 2 is to perform mouse studies. So we'll be using these compounds in our mouse models of Duchenne muscular dystrophy and asking whether or not we can help muscle regeneration in these mice, as well as improve muscle force. And so I'd like to thank Defeat Duchenne Canada for all their support. Thank you for your support and for listening.
Our next video is from Dr. Eric Hoffman, who is the president and CEO of Reveragen Biofarm in Rockville, Maryland, US. And this project is in its last and final year of grant. And this is Defeat Duchenne Canada's first clinical trial support under a venture philanthropy model. And I just wanted to say a few words about steroids before we hear from Eric. Steroids are the current standard of care for Duchenne, and it's a medication that is able to delay the loss of walking, or at least it's demonstrated that it's been able to delay the loss of walking. However, long-term use of high-dose steroids comes with a long list of side effects. Some of those are short stature, bone loss, an increase in risk of fractures, behavioral issues, and weight gain. And a lot of families who are listening completely live and breathe this every day and understand this. And that's just a few of the side effects. Vamorolone is an exciting steroid alternative currently under investigation by ReveraGen under the leadership of Dr. Eric Hoffman, with the hopes of having the same benefits as current steroid treatments, but with less side effects. So let's hear from Dr. Hoffman next.
Good afternoon. My name is Eric Hoffman. I'm the co-founder and CEO of ReveraGen Biopharma in Rockville, Maryland. And ReveraGen is the sponsor of developing vamorolone, a replacement for corticosteroids in treatment of Duchenne muscular dystrophy, currently under development. We received a $1 million contract award with Jesse's Journey, now Defeat Duchenne Canada, in 2020, so 2 years ago. And this was to carry out a clinical trial of an expanded age range in Duchenne. So all our previous trials of vamorolone have been in 4 to 7-year-old steroid-naive boys, and these have been carried out in 33 sites in 11 countries over the last couple of years. But this trial is a Canada-only study, so it's a clinical trial enrolling only in Canada, and it's to, for the first time, study younger 2 to 4-year-old patients and older 7 to 18-year-old DMD boys. And the older boys can be both previously treated with corticosteroids, so switch from corticosteroids to vamorolone in the trial, or have been off of corticosteroids for 3 months or longer. So this $1 million award is a large award, and it was done in a different model, which is sometimes called venture philanthropy, which means it's not so much a grant as a agreement or contract between Jesse's Journey or Defeat Duchenne Canada and ReveraGen as a drug development company, where it's a shared risk, shared benefit model, where based on later drug sales of vamorolone, Jesse's Journey will be repaid by ReveraGen about up to 400% or fourfold more than the grant was. So if vamorolone is successful and gets approved and sold, those drug sales will repay Jesse's Journey and repay up to fourfold their investment over the last 2 years. So where do we stand with this trial? So the trial has been going quite well. It initiated, it's actively enrolling it. It's, first of all, I should mention that the study chair is Jean Ma at Calgary, a wonderful and expert physician scientist who has participated in all the vamorolone trials to date. And so she is the medical clinical lead for this Canada-only study. The study is currently actively recruiting at 3 sites in Canada that are open, and that's Dr. Ma in Calgary, Dr. Selby in Vancouver, and Dr. Lockmuller in Ottawa. And we expect to begin recruitment very soon, within the next few weeks, at 2 additional sites, with Dr. McMillan in Montreal and Dr. Gonoraski in Toronto. To date, even though the trial has been actively recruiting at just 3 sites for just 2 or 3 months, we are already reached 30% enrollment. The entire trial is 44 participants, 20 in the younger age group, 24 in the older age group. And so the trial has been recruiting very briskly, and there seems to be enthusiasm of patients and families and physicians for joining the trial. And this is a very important trial because it really is the first time to expand the age range of testing of vamorolone. One of the hopes is that with less side effects like the loss of stunting of growth, which we see, and loss of bone morbidities, and perhaps other side effects that you see with corticosteroids, it becomes more feasible to treat younger and younger boys, and that might have greater and greater effect long term. So that's why one of the reasons this trial is so important. And also it's important for the first time to study the transition from deflazacort to vamorolone or prednisone to vamorolone. We also studied in a double-blind trial, but not deflazacort. So this trial is very important for multiple reasons. Again, this is a Canada-only study, which is also unique, and it's really because of the relationship with Defeat Duchenne Canada that it was agreed to be a Canada-only study, in part because we expected there to be a lot of demand for entering the trial internationally, and we didn't have the bandwidth as a company or the number of slots to make it possible to enroll in many countries. So it really made sense to limit the number of sites to meet the recruitment needs. And it made sense then with this relationship with the Defeat Duchenne Canada to just limit it to Canada and just the 5 sites there. And as I mentioned, in just 3 months, we're already 30% enrolled. So that thinking or that rationale seems to have been quite accurate and appropriate. The next steps are to complete enrollment and activate the last 2 sites. And so we expect the enrollment to take about a year. And you should contact the sites if anybody's interested, and then to seek regulatory approval. The situation for regulatory approval is in the US, so you go through the FDA, and we're completing the FDA. It's already been initiated within the next month. So we anticipate that the full NDA will be submitted by the end of June, and that's in collaboration with Synthera Pharmaceuticals, who's the marketing partner. And that could, if it's reviewed well by FDA, then lead to drug approval in about 7 to 12 months. In parallel, we'll seek regulatory approval based on existing data with both EMA through Europe and with Health Canada in Canada. I should also mention that this current trial with that Jesse's Journey and Defeat Duchenne Canada is funding through Revirigen. The trial management is expected to transition to Synthera within the next couple months, but the agreement and the contract resides just with Revirigen. So Revirigen and Defeat Duchenne Canada will maintain this relationship, even though the trial management and the sponsorship will shift to Synthera. And all have agreed to this, including the Defeat Duchenne Canada board. Thank you very much for your support. This trial would not have been possible without your support and also your participation in clinical trials. Clearly, drug development could not take place without the families and the patients' participation in these critical clinical trials. We certainly recognize they are a major demand on time and resources of the families, and we are very grateful and humbled by the energy of the families and the willingness to participate in these trials. It's really a collaboration not only between us and Defeat Duchenne Canada, but with all the families in Canada, particularly those who have participated in the trials, which have been quite a few over the last few years. Again, thank you for your support and have a good day.
And I see we have a question in the chat. It says, is there a chance to ask a question about the research update? Can it be clarified whether Dr. Sato's chronic treatment study is taking place in human patients? We are very fortunate to, I believe we have Dr. Sato in the audience. So I don't know if Dr. Sato is comfortable answering that question in the chat box. I will say that the in-human trials for Dr. Sato Sado's investigative oral therapy is beyond the scope of the current grant research. So first we need to, see the success of this first stage before it can go into human trials. But Dr. Sado, please feel free to answer that question in the chat if you're able to. So thank you. And just a reminder to our audience members, please continue to put your questions in the chat, and there's also the Q&A below if you want to use that. So with that, we have our last and final grant is from Dr. Duchenne Duan at the University of Missouri. And sorry, let me advance the slides. Oh, that's not working. Oh, there we go. He is the last grant that's in its third and final year of funding. And Dr. Duan and his team are developing the next generation AAV vectors. And these are essentially the vehicles that transport gene therapies into patients. Gene therapy offers a lot of hope for our community, but there's still much work to ensure that these therapies are both safe and effective. And Dr. Duan is currently looking to address this. Unfortunately, we do not have a video from Dr. Duan, but if you want to learn more about his research, you can certainly go on our website under our research grants to learn more. So with that, I would now like to pass it over to Dr. Lisa Hoffman, who's now going to announce our new research award recipients for 2022. Dr. Lisa Hoffman has been a longtime volunteer for Jesse's Journey, now Defeat Duchenne Canada. She's been part of our research committee since 2012, and she has chaired our research funding advisory committee for 10 years now. Lisa has been a fantastic partner with me over the last 3 years in my short stint here at Duchenne Canada, and it's been an absolute pleasure. So thank you, Lisa. The floor is now yours.
Well, thank you so much, Nicola, and the pleasure is also mine. It's been a pleasure to work for the foundation, the Defeat Duchenne Canada. So again, this is just a great organization. So now I'm pleased to bring up our 2022 research awards. We received 18 applications from India, Japan, Canada, the UK, the USA, and Uruguay. The total amount funded in 2022 being $1.14 million. So it's now my great pleasure to introduce the first of our 2022 awardees, Dr. Lewis Kunkel, coming from Boston Children's Hospital, Boston, Massachusetts, USA. Dr. Kunkel and his team proposes to promote the proliferation of skeletal muscle cytolate cells, modulating the Notch3 signaling pathway, which is involved in the regulation. This approach is aimed at restoring dystrophic muscle in DMD patients, regardless of their gene mutation. And with that, Dr. Kunkel.
Thank you. Thank you, Lisa. And I think I'll share my screen. And pull up my proposal. Can you see my screen?
Yes, you're good.
So I'm going to tell you a story that's a combination of efforts from my lab and the lab of Mayanna Zatz, who is pictured here. It's going to take place via a postdoctoral fellow who we jointly have, Felipe de Saluzzi Lite. So I've had a long-standing interaction with Mayanna Zatz in Brazil over many, many years. And a few years ago, Mayanna brought a project to my lab concerning a series of unusual golden retriever dogs who escape dystrophin deficiency. And so a postdoc from her group came to my lab, and we did genetic analysis to try to determine what gene might be involved in influencing the outcome in these golden retriever dogs. And I might add that these dogs without dystrophin lived a completely normal 12-year life. So we've analyzed these dogs and found that what was happening is they had a variant, a gene variant in a gene called JAG1. And it's one of the ligands for the Notch pathway. And we were able to show that the increased expression of JAG1 in our zebrafish model of Duchenne dystrophy completely corrected, or very close to corrected, the disease status of these fish. And you can see this is normal muscle. I mean, this is abnormal muscle. This is muscle that's been corrected by the overexpression of JAG1. So my lab has been working with JAG1. And as we went along, Mayanna had been working with a number of other interesting patients. So are there human patients who have similar outcomes to dystrophin deficiency as our golden retriever, as the golden retriever dogs? And so Mayanna is a clinical neurologist in Brazil, and she sees numerous patients. And the first set of patients she had were two half-brothers, both of which had an inactivating mutation in their dystrophin gene. The younger boy had onset at age 3, had a DMD diagnosis, was established by molecular analysis at age 7, and was in a wheelchair by age 9. He has no dystrophin, as shown here. His brother also had no dystrophin, but he was able to climb stairs without assistance, walk normally, stand up without assistance. He's currently still, he drove a car until recently, but he had a few difficulties. Mayanna also searched the literature and found a second family where there was a second person in Spain who also had a very, very mild phenotype, whereas the other family members had a more severe phenotype. He was able to walk at 24 years, climb stairs, and didn't lose the inability to walk until he was 30. So Felipe and Mayanna did a very, very large scale screen, just like we did with the dog, and essentially asked by whole gene exome sequencing, RNA-seq, proteomics, and family history, could they figure out what genes were in common between these two escaper humans now that might explain why they escaped this phenomena. And to make a long story very short and multiple years of progress, we were able to identify a single gene that had a variant in both patients in both the same domain of the protein. This is actually unpublished information, so I want everybody to keep that in mind. The gene involved is Notch3. Both patients had a variant which was known to have an inactivating or a loss of function, or decrease in function in the same Notch3 domain. Well, Notch3 is the receptor for the ligand JAG1. So now we've put together JAG1 variant in dog escaping, and the humans that are escaping or showing milder phenotype have variants in their Notch3 receptor. This implies, since Notch3 is highly expressed in stem cells, that this Notch3 pathway is very important to the outcome in patients with dystrophin deficiency. So what do we know about the variants? They're not pathogenic. We know they have a cellular phenotype. They change function. We've done functional studies with them. And muscle progenitor cells with these variants proliferate more than controls or other patient samples. So what do we plan to do? What is our proposal to the Defeat Duchenne Canada? Well, first we want to understand the Notch pathway downstream of these variants. We want to regulate Notch3 expression in our animal models. So we have zebrafish that have dystrophin deficiency, severe muscle phenotype, and we have mice that have severe phenotype. So we can actually express the variant Notch3 genes in the presence of these other these dystrophin deficient animal models. We're going to look for genetic modifiers of this. And we also know that there are drugs that influence the Notch3 pathway, and we're going to test those in our animal models. We are also creating mice who have one of the variants in the Notch3 gene, which happens to be in a very conserved domain. So mice have the same amino acid sequence at that point as do humans. And we're making the variant there. And that's being done as a takeout at a place where it's done commercially. And we're going to have mice very soon that are carrying the Notch3 Spanish candidate variant. We will then cross those mice with our MDX mice and ask, do we still see the same phenotype outcome? In general, our whole proposal is to study how the Notch3 pathway influences the outcome in both our animal models and how we can modulate it to be corrective in muscular dystrophy. And I'm willing to take questions from the audience on that.
Great talk, Dr. Kunkel. Let me just take a look here to see if there's any questions popping up. Maybe Nicola has come across some.
I don't see anything in the chat or the Q&A at this time, but we can keep going, and we may have some questions towards the end.
Okay, thank you so much, Dr. Kunkel.
You're welcome.
Let me just get there we go. So it's with my great pleasure to introduce our next speaker of the evening, Dr. Michael Rudnicki from Ottawa Hospital Research Institute in Ottawa, Ontario, Canada. Dr. Rudnicki and his team propose to use exosomes to systemically deliver Wnt7a for the treatment of DMD. The ability to harness Wnt7a signaling to ameliorate DMD pathology is a potentially major advance towards therapeutics. And with that, Dr. Rudnicki.
Thank you very much. Let me share my screen. Okay, how are we? Can you see all this? Okay, let me first begin, like the others, thanking Defeat Duchenne for their support. I mean, having Defeat Duchenne support these kinds of high risk, high gain projects with a translational arrow really helps propel the field forward in really an important way. And their support over the years has really allowed us to do a lot of interesting things. Emerging data from multiple labs, including myself and also Lewis Kunkel's lab, strongly support the notion that DMD is a stem cell disease. Not only do we have this fiber issue, but the reduced ability, this impaired ability to repair the muscle damage makes DMD into its very progressive form of the disease. And so we're exploring different avenues to mobilize muscle stem cells and stimulate the regeneration process. And the grant from Fighting Duchenne is pursuing one of those avenues that they're quite excited about. And this is exploring how to deliver Wnt7a systemically. So what is Wnt7a? It's a secreted protein that is normally highly upregulated when a skeletal muscle is damaged. Unfortunately, it's not upregulated when it's chronically injured, like in DMD. It's off. But when you acutely injure the muscle, it comes on quite strongly, and it does multiple things. It expands the stem cell compartment, it stimulates the dispersal of the cells, and activates the growth of the muscle fiber, and all of this together makes stronger, better muscle. And we showed that by injecting Wnt7a directly into skeletal muscle in mice with muscular dystrophy, we profoundly ameliorated disease progression. These muscles were stronger histologically, they looked almost normal. It really had a phenomenal effect. The problem with Wnt7a, though, is that it's a highly insoluble protein and you can't deliver it into the blood circulation. It doesn't go anywhere. So it would have to be injected locally, and that makes it a really intractable as a treatment. So our understanding of Wnt signaling, there's multiple different types of Wnts with all kinds of different roles in different cells. But the dogma has been is that Wnt7a is secreted as free protein. There's a sender cell. It will diffuse over a short distance to a receiver cell where it binds a receptor on the surface of that cell, shown here in green. This is a member of the frizzled family, and there's multiple frizzled receptors. And then a particular cellular response will occur. And all kinds of different roles are played by Wnt signaling through embryonic development and in tissue regeneration. And it really depends on the cell type we're talking about. One of the questions we set out to address was whether or not Wnt7a was in fact not secreted as free protein normally, but was secreted in what are called exosomes, or extracellular vesicles is another name. And so these are small packets, membrane-wrapped, tiny vesicles that would display the Wnt protein on their cell surface, and they could either interact with the receptor via that or be absorbed into the cell. Now, why exosomes are interesting is that they can deliver all kinds of materials over long distances of the bodies between organs and so on, because they can travel through the circulation. So we wanted to establish, first of all, whether or not Wnt7a is expressed on exosomes and whether or not it could be used in a therapeutic way to treat DMD. Now, first of all, this just shows you how Wnt7a is normally off, and in mice with muscular dystrophy, we don't see expression either. Whereas if we do an acute injury with cardiotoxin, you can see high upregulation of Wnt7a in green in the newly formed muscle fibers, where it promotes in a feed-forward way the regenerative ability of muscle. And if we add more Wnt7a, you get bigger, better muscle. So it's really a feed-forward mechanism that promotes and augments and accelerates muscle regeneration. So if we look via electron microscopy at these regenerating muscles, we can see what are called multivesicular bodies where exosomes are being manufactured, and by immunostaining, we can see Wnt7a on the surface of these exosomes. And if we go zoom in, here we can see what look like exosomes with Wnt7a on them. So we could purify these using transflow filtration, and we used a mouse where we either deleted Wnt7a or kept Wnt7a. And you can see with these purified exosomes, we can see by electron microscopy and the staining for Wnt7a, we can see nice expression. But if we delete it, we have zero expression. And I'm sorry, I went the wrong way. And if we then treat primary myotubes, muscle fibers with this, one of the responses we see is growth of the fiber. We get these big fat ones. We can also see this in vivo, but this is a nice fast test in tissue culture for activity. So recombinant protein gives us this nice growth. Exosomes within Wnt7a give us this nice growth, and exosomes without Wnt7a, not so much. And we can see this nice enhancement of exosomes loaded with Wnt7a that seem to work better than an equivalent amount of recombinant protein. So we also did some science along the way. We were really interested in understanding what is it about where in Wnt7a was the sequence required for attaching these Wnt7a to exosomes on the surface of exosomes. And through a whole series of constructs where we made all kinds of changes, we identified a small 17 amino acid portion of the protein. This is Wnt7a here. It's like a C clamp. It binds the receptor in this hole here and activates the signaling response. But this small sequence here is required to bind to exosomes. It's a 17 amino acids piece, very, very small, between two prolanes, unstructured motif. And this is required for exosomes. If you mutate it, you lose exosomal secretion and you have enhanced secretion as free protein. Also, we can take this 17 amino acid, link it on a protein that's normally not on exosomes, and direct it for exosomal secretion. So this is an experiment where we took HALO, which is a protein originally from bacteria, doesn't load on exosomes. We can link it to the ESP and it loads on exosomes. And what HALO does is it binds a dye that when we purify exosomes from producing cells of these two different sorts and then put them on cells, the cells can take it up because exosomes deliver cargo to cells over long distances. And then we can stain for HALO in that recipient cell. Whereas if you don't have the ESP, there's no HALO on those exosomes and nothing is taken up. So this really represents a very nice proof that this is a very useful tag that we can use to engineer proteins so that can be targeted to exosomes. And we're going to use that in our grant application to enhance specificity for muscle and muscle stem cells. Through a whole series of experiments that I'm not going to talk about, we defined which of the amino acids are involved with binding to a particular protein that we now have shown is on the surface of exosomes. These are codomer proteins, in this case, COP beta 2 protein. These are proteins that weren't understood previously to be on the surface of exosomes. And this acts as it looks like a donut, which the Win7a then is presented facing the outside on the surface of exosomes. It's a very cool story. So we can take these exosomes loaded with Win7a versus exosomes without Win7a, inject this into the blood system of mice, and then detect where these proteins go. And interestingly, exosomes with Win7a displayed over a twofold enhancement of targeting to skeletal muscle over exosomes without Win7a, suggesting that Win7a itself is enhancing targeting, but it's getting to skeletal muscle. We then built a system where we could have an enhanced mouse that had an enhanced sensitivity to Win7a because it lacked Win7a. So this is an MDX mouse with Duchenne muscular dystrophy in which Win7a has been deleted. We damage the muscle, we do a single tail vein injection of Win7a loaded on exosomes, and then at the end of 21 days, we do forced generation experiments, histology, and so on. And I'm showing you the forced generation experiment, and you can see this remarkable enhancement in forced generation because we've stimulated regeneration. Really quite phenomenal from a single injection. So of course, now what we want to do is do this in mice that have their Win7a. It's normally not upregulated because we think that it should be able to have a therapeutic effect and reach all the muscles of the body. And the reason why we think that, in these same mice, without an acute injury, we looked at the diaphragm, and the mice that were treated with Win7a had a diaphragm that looked largely normal. It didn't have these big damaged areas that we see in the mice that were treated with exosomes lacking Win7a. And this muscle, as I said, was not damaged with cardiotoxin. It's just undergoing the normal injury that occurs in DMD. And so this strongly suggests to us that we're going to be able to hit all the muscles in the body. And those experiments now are about to be initiated. So these are objectives in progress. We're engineering an enhanced exosome to deliver Win7a that'll enhance both targeting to muscle tissue and to muscle stem cells, and will have an enhanced activity. We're going to treat dystrophic mice long-term and ask what it does over the life of the animal. And we're also going to evaluate what happens when we combine exosomal treatment with gene therapy. And then we're also going to test and optimize a formulation for activity on humanized muscle tissue. These are the people who did all of the work. And most importantly, Uxia Guerrien Rodriguez, a Spanish postdoc, led the project. And she had significant help from David Datsku, who helped identify the protein on exosomes using a mass spectrometer mass spec kind of approach. And of course, Defeat Duchenne, I thank you. As Ronnie said, your support has really been essential to move this project to the next level. I put Jesse's Journey logo here because I'm very fond of that name. I knew Jesse Davidson and of course his father who established what's now called Defeat Duchenne. I think your leadership in this space has really been phenomenal. And I should disclose that I'm a founder and chief scientific officer of a company called Satellos Bioscience. The intellectual property has been licensed to Satellos concerning Win7a, and together with Satellos, we're developing alternative small molecule approaches also to enhance and stimulate regeneration. Thank you very much.
Great talk, Dr. Rudnicki. Any questions? Let's see here.
We have a question in the chat, Lisa.
Okay.
Can you see that, or?
Is that from Matthew Hendry?
Yes.
Okay, so the question is, so if I'm correct on this, a lot of this really is next level medicine, a Kennedy Khrushchev of medical advancement, if you will, instead of doing for the body, the drug being used is speaking to the body?
Yes, I think several of the talks that you've heard today concerned with enhancing intrinsic processes to, and these will be regenerative medicine approaches to enhance and restore the regenerative ability of muscle stem cells in DMD patients to normal levels. And that we believe that restoration of regeneration activity will potentially be, I don't want to use the C word, but curative. I mean, Lewis Kunkel's dogs suggest that if via the jagged 1 mechanism or the notch 3 mechanism, we really have a profound effect on the progression of the disease and delay it markedly. The dogs with the jagged 1 mutation have a normal lifespan and a normal fourth generation of muscle and don't have the cardiac effects. It's quite remarkable. I think this is a new paradigm. It's a paradigm shift in our approach to treating DMD, and is I'm really quite excited about.
Thank you very much. Any other questions for Dr. Rudnicki? Okay, with that, we'll move on to the final speaker of the evening. And if there's more questions at the end, we will have a few minutes that we can have some more asked and answered. So with that, it's my pleasure to introduce Dr. Guy Odom from the University of Washington, Seattle, Washington, USA. Dr. Odom and his team proposed to redesign microdystrophin for recombinant adeno-associated viral vectors, or AAVs, without immunogenic epitopes that could be recognized as foreign by the patient's immune system. Therefore, if successful, this project could overcome current limits and issues of gene therapy. And with that, Dr. Odom.
All right, let me share the screen. Can you see me now?
All good, thank you.
Okay, great. Thanks very much. I just can't say how much of an honor, privilege it is to be speaking here today with such a great organization and such great scientists contributing to this very noble cause. Let me, I'm gonna, by the title, gonna jump into aspects of gene therapy vectors. So I'm going to chat a little bit about immune responses to dystrophin, both a former trial and current trials. Early trial, which preliminary data that I'll be presenting here that was directed by Jerry Mandel, found anti-microdystrophin T-cell responses in 1 of 6 patients. And in current trials by Pfizer, Sarepta, it was noted that there were some serious adverse events in 5 patients and was mapped to epitopes in exons 8 to 11 in all of those patients. So this is just a schematic of the microdystrophins that have been in and are currently in clinical trials. They're all very similar. With the exclusion of the early trial, they carry muscle-specific promoters, have various spectrin-like repeats. They both include the internal actin binding domain and the cysteine-rich domain that's critical for nucleating the dystrophin glycoprotein complex. And so here you can see it's just mapped out where you have that region of exon 6 through 8 that were found to pop up in the current trials with the SAEs that were reported. And to combat that, for our approach here, we're looking at a process referred to as de-immunization. It's a relatively recent technology for the location and removal of T-cell epitopes, and it combines immunological databases, epitope databases, molecular and computational techniques, where if you can either predict or know an epitope that you want to detarget from recognition of the T-cell receptor, you can quite frankly do that. There are 2 drugs currently in trials now that have demonstrated that. The machine learning predictions, generally there's a panel of HLAs. Oops, sorry, skipped ahead there. Sensitive mouse. So you have a representative set of 8 to 14 HLA alleles, and each allele has a specific structure that it allows it to accommodate a peptide that's presented by to the T-cell receptor from the MHC presenting cell. And so this is the former trial, the Mandel trial. We're looking at this quite closely and collaborating with David Baker's lab, who is heavily involved in protein design, director of the Institute of Protein Design at the UW. We presented the possibility of de-immunizing some of the epitopes that were discovered in the early trial. So that's shown here with the light blue is the P17 epitope, P19 epitope is in pink, and then the yellow is the P23 epitope. And they were all represented in exons 6, 7, and 8. So in that similar region that was found in the current trials. So question, can we de-immunize microdystrophin and eliminate T-cell responses without destroying functionality? So collaborating with the Baker lab, David's group generated 3 designs for the P17 epitope represented in panel A, where we have DES1, DES2, and DES3. We subcloned those into our Hinge3 microdystrophin and generated vector, injected MDX mice, aged the mice out to 8 months and evaluated protein expression. You can see the immunostaining of the native Hinge3 microdystrophin. It has bright green staining detecting dystrophin. And as well for the 3 designs, there was an intermediate performance for DES3 actually, where the H&E looks quite well. DES2 was our best candidate for this small panel, and DES1 was a complete failure basically. But we also did in situ physiology and gathered data on specific force, maximal force production, and we also did an injury assay. This was for the gastrocnemius muscle that's shown above in the staining panel. And we also did recovery force production. Moving to the second epitope that was identified in that trial, the P19, we generated 5 designs shown at the upper left relative to the wild type. And we tested those in MDX cultures shown right below to look at relative protein expression. We then went into MDX mice again, showed expression at on the sarcolemma, they're shown in green. And we performed a hind limb torque assay of the various candidates and got some good encouraging results with the black line is wild type, and then the red line is the native microdystrophin, and the blue line is one of our leading candidates, the P19 DES2. So from there, we also combined these 2 epitopes to look at, since they were in such a close proximity to one another and also adjacent to an actin binding site within the actin binding domain, we wanted to confirm that that they would work together in a good way. And we also evaluated heart by cardiac MRI, and we would see close to normalized ejection fraction, stroke volume, and then again we did the hind limb assay, and those showed recovered specific force production and protection from contraction-induced injury. So are the redesigns human DMD epitopes de-immunized? That's the big money question, so to speak. So yes, they are. For those 2 epitopes, we have T cells from the original patients that were run on ELISpot assays. And you can see in A, we have the gamma interferon response from those T cells that are ablated in design 1, 2, and 3. We have our PMA positive control and our P17 native that was nicely reproduced from the Mandel New England Journal of Medicine paper. And similar results, but not identical with the B19 epitope. We had a slight flare up with the M1 design, but 4 out of the 5 designs were shown to be de-immunized. So that was very encouraging. So in thinking about this development, we've also had our lab has had a great interest in Utrophin as a biotherapeutic. And as I'm sure you're aware, that there's several papers recently that have come out involving gene editing for upregulating endogenous Utrophin, and also Kay Davies has been heavily involved in this sector for many years in terms of generating compounds to upregulate Utrophin. And as well, there's been a recent dog model paper that showed that microdystrophin was immunogenic in that dog model. It was a German short-haired pointer model. It was a complete deletion null animal. And while the microutrophin did not present with an immune response in that model system. And then there's a paper we published back in 2020 that showed therapeutic differences in fiber types with our microutrophin. And so from there, we want to see if we can improve the function of microutrophin. And so we're making some changes to the structure, including varispectrin repeats. We want to bring in centrophin binding domain from the C terminus to see if that can recruit additional players within the DGC to provide for more stability. And also we want to de-immunize the dystrophin repeats 16 and 17 to endow microutrophin with the ability to recruit nNOS to the muscle membrane. So here's an example of some of the redesigns we've created with repeats 16 and 17. A shows the native structure, and then B, C, and D are 3 of our designs. We're adding more to this arsenal based on the system that we're developing here for our grant. So shift gears a little bit. This testing system involves a high degree of collaboration with the laboratory of David Mack, where they painstakingly gone through many protocols to drive iPSCs or into muscle or myogenic programming. So here this is over the time span of 45 days, where you start at day 0, and then at where you have the 2D myotubes at the end. That's over 45 days, but around 14 days, you start to see skeletal muscle markers, alpha, actinin, titin, desmin, and dystrophin displayed here, and the key culture conditions that are displayed by the color boxes. So from there, also working with David and Nate Sendecki, this is part of a UW startup. Curie Bio developed the system to essentially as a drug screening type of scenario, where you have engineered heart tissues or skeletal tissues, and you develop a 3D tissue in the dish. And so you have your engineered heart tissue, or EHT, in at the base of the well. And then there are 2 posts that are attached. One's a rigid post and one's a flexible post. And so you're able to measure contraction. And so that's represented in C. And then in D, we have a calcium response. And then in E, we have random rhythms that are created. And in F, we're showing the ability of AAV, in this case, AAV6M cherry, to express throughout the 3D culture. So just to kind of summarize the specific aims, this is a 3-year period, and it more or less overlaps slightly, but aim 1 will be covered in the first year, and we'll be evaluating various vector systems within the 3D culture model. And in parallel, we'll be creating the de-immunized dystrophin repeats of 16 and 17 candidates that can transition into our micro-utrophins that are being evaluated in aim 2. So the utrophin vectors will be tested functionally within the 3D DMD tissues. We're also going to look at MHC-associated peptide proteomics to give us insight in terms of how these redesigns respond to human antigen-presenting cells. And so we're looking forward to working with the Benza on that portion of the project. And aim 3 really is moving to the MDX rat model, which we believe better recapitulates the human condition. And so we'll be doing extensive functional measures, hind limb and cardiac, also looking at various assays for respiratory function, mitochondria metabolomics assays, and histological assessment. So with that, I have the acknowledgements. We have a strong intertwined group here at the UW that specializes in muscle biology, including David Mack, Stephen Hoshka, Jeff Chamberlain, Julie Crudell, who worked up the ELISpot data for us. We also have the group with David Baker's lab we're highly collaborative with, who actually spin off from his lab with Cyrus Biotech. And so they're also carrying the torch for de-immunization. And we could not have done the T-cell work without cooperation with Ohio State and Chris Walker. And of course, the Seattle Wellstone Vector Core, which has the vector expertise to carry that torch. So I'd like to thank you for your attention, and I'll be happy to answer any questions.
Thank you very much, Dr. Odom. A great presentation. So there is a question for you. Are those machine learning prediction sequences similar to the mRNA sequences, essentially computer technology generated sequencing?
I'm sorry, I'm not familiar with the latter technology. Not sure I caught that.
And I'm not familiar with that either. Perhaps Matthew can.
I would say no, that it's based on Rosetta, and Rosetta developed through the Baker Lab, and the de-immunization aspect had to do with integrating immune epitope databases and modeling from that standpoint.
Great. Well, are there any other questions? Let me just take a quick look. Doesn't appear to be. So with that, I think I would like to thank all three presenters again for their excellent presentations. Your findings are extremely encouraging, very exciting. So thank you very much, and continue with all your hard work.
Thank you so much, Lisa, and congratulations to all our new grant recipients. We really look forward to hearing more about your progress in the coming years. And I just wanted to, again, just highlight our research highlights for this year. If we can go to the next slide, Rochelle. Thank you. So in our 27 years of research here at Jesse's Journey, now Defeat Duchenne Canada, including the grants that we're funding this year, we are granting $16 million towards research projects for Duchenne muscular dystrophy. And we've proudly funded now 56 Duchenne research projects from all over the world. And that's a fantastic milestone for our organization as we continue to support this research. Sorry, I'm just going to go to the next slide. There we go. Research is the road to hope for Duchenne muscular dystrophy. And while science is the method behind our mission, there would be no progress without people like you. It's donations from our community and the stories behind them are the only reasons we have been able to fund promising research like you've heard here today. And I would like to announce before we close it off is our founding family, the Davidsons, have been the living examples of how far parents will go to fight for the lives of their children. This spring, in memory of their son Jesse, and in honor of all children and young adults living with Duchenne, the Davidsons have agreed to match the first 50 new monthly donations until the end of the year, and match the monthly increases of current donors who choose to amp up their support. And you can sign up now to be a monthly giving champion at defeatduchenne.ca/champion. So that completes our research announcements for 2022. The recording from tonight's event will be emailed to you later this week, along with the press release, and we hope that you'll share that with your network. Ensure you receive our community updates by subscribing to our e-newsletter at defeatduchenne.ca. So with that, I thank you all for joining us tonight, and join the conversation as we defeat Duchenne across Canada by following us on social at Defeat Duchenne. Thank you very much, everyone, and have a lovely evening.
Take care, everyone. Have a great night.
Thank you. Oh, I see we have a question. Any questions in there, we'll be sure to follow up with you. So if you put a question in the Q&A, we'll follow up after this meeting. So thank you very much, everybody.
Bye.
Goodbye now.